Okay, so this is going to be a review of respiration, including the Krebs cycle, glycolysis, the mitochondrion, and the process of using the electron transport chain to make ATP. Really only got one shot at this, so hopefully the volume comes through clear enough. We're going to start off by giving ourselves a diagram of the mitochondria and talking about what happens and where, and then we'll come back to that and look at what's going on in the electron transport chain as it relates. So to start with, let's draw our mitochondrion. The mitochondrion is kind of a bean-shaped organelle. It has two different membranes. It has an inner membrane and it has an outer membrane. I've already made this a little bit less than ideal. That's alright, all we're doing is looking for something that we can use. So, the mitochondrion has an inner membrane, an outer membrane. Outside of it would be the cytosol, or the cytoplasm. Inside is the matrix. There is the outer membrane. There is an inner membrane. And there is what's called the intermembrane space. Now, that intermembrane space is all of this area right here in between the inner and outer membrane. This is the intermembrane space. So this definitely separate from the cytoplasm and cytosol there, the intermembrane space. Now the processes that occur in all of these areas are important to note. So the processes I'm going to put in a different color. In the matrix we have the Krebs cycle occurring. Out in the cytoplasm we have glycolysis. And we have fermentation. On the inner membrane, we have something called the electron transport chain. While not a process necessarily, the electron transport chain is a really important part. So the electron transport chain, I'm just going to label as the ETC on this membrane. Now, when you think of what's going on in respiration, it helps for me to visualize it like water. So water that falls from a really high distance has a lot of energy. At the bottom of a waterfall, it'll cause a lot of erosion. So if you question how would I take the energy that we have in a waterfall and actually utilize that, well, what I'm going to do is think about a waterfall coming over the top. It causes a lot of erosion down on the bottom of that waterfall, and that erosion is energy that's lost. The same happens when you have combustion, which is basically, it's the same equation as respiration, but it happens out of control. It happens kind of in one big massive step, if you will. All the bonds are broken, all the energy is released as light and as heat, so it gets really hot. Well, the human body stays down at like 98.6 because we manage that process of breaking bonds. If we wanted to extract energy from this waterfall, you could put a water wheel up here, and you could let that water wheel turn and gain some mechanical energy. You could put another one down below that, and you could continue this process down the waterfall, stealing some of the energy at each spot And in the end, you could actually use up most of the energy and reduce your erosion down here because the energy wouldn't be lost. It would be gathered, it would be taken and used. Now, in a electron transport chain, what we're doing is we're actually using the inner membrane of the mitochondrion, and we use these things called proton pumps. Now, I will be the first to admit, they look nothing like what I'm drawing right now. What I'm drawing right now is made to look more like a water wheel, to make it make some intuitive sense, not because it's anatomically accurate. It is not anatomically accurate. But it helps you understand what's happening more easily than the way that they actually look. Our chemistry isn't to the point that it would need to be to use the way they actually look for understanding. So this would essentially be our electron transport chain. Now you have different chemicals that carry electrons in here, and they set them down on this electron transport chain. And the electrons that are over here want to get over here. The electrons once here want to get over here. Once they're here, they want to get over here. But they can only get to here from this one. They can only get to this one from this one. So what happens is these two different molecules, the first one is NADH. This molecule comes in and it breaks down into NAD plus and H plus. In the process, it gives off two high energy electrons. So these two high energy electrons, when they get to here, they want to be over here. When they get here, they want to be over here. These objects let them do that, but in the process, the only way that they'll allow that electron to go from here to here is if that electron, say, turns this to pump an H plus to this side of the membrane. And then to get from here to here, it has to pump an H plus to this side of the membrane. Same thing here, same thing here, same thing here, same thing here. Now, once this electron has gotten all the way to the end of this electron transport chain, it's got no energy left. So what happens is that electron is joined to an oxygen plus 2 H plus 2 electrons. And what that gives us is when we put it all together, this equals H2O. Now, H2O accepts those and can be expelled from the body. So basically the food that we use, after it goes through after the energy from it goes through the electron transport chain, it's eliminated as water. Now, these hydrogens down here are just the ones that came from NADH. There is another molecule in our bodies that also carries electrons from the Krebs cycle. That chemical is FADH2. FADH2's electrons don't have as much energy, so it doesn't drop them off as high, but it breaks down into FAD plus 2 H plus 2 electrons. And these 2 electrons, they're not up here. Those electrons are actually released right here. And they do the same thing. It wants to get here once it's released in this spot, but to do that, it has to pump an H plus. Same thing, once it's here, it wants to get over here. Once it's there, it had to pump an H plus to get there. Once it's here, it wants to get over here. Once it's here, it wants to get over here. And again, it ends up making water. And each time it goes through one of these, it pumps an H plus. Now notice it didn't pump as much H plus as the NADH. So what happens is it's worth slightly less energy. Now, the way that energy is made, the energy that we care about, our whole goal, and what the mitochondrion really does, is it converts sugars into ATP. Now, these ATPs are made on another enzyme that's over here, and it looks nothing like what I'm about to draw. But again, this makes more sense shape-wise than the way it actually looks. Now, this would be a chemical an enzyme that's pretty accurately named. It's named ATP. This thing is ATP. ATP synthase. Reaching so far to write this, my writing's getting awful. What it does is it grabs an ADP and a phosphate. These hydrogens have built up over here, making this really positive, so they don't want to be here anymore. They want to get back through. For them to get back through, they have to shove this over, slamming the ADP, or adenosine diphosphate, into another phosphate, and when they slam them together, you end up getting ATP. This is the high energy source that your enzymes use, and that's the whole reason why we use cellular respiration. But ATP is not storable, so we have to constantly break down fats and sugars or proteins to make ATP. The electron transport chain allows us to use those extra electrons that are in carbon-carbon bonds and carbon-hydrogen bonds. It lets those oxidize down to H2O, and in the process, the NADH carried them over here, put them on here, and in the process of them becoming more oxidized and landing eventually on H2O, they pumped this hydrogen. When the hydrogen wanted out, it actually made ATP for us through ATP synthase. Now, how does this fit into this? Everything that's drawn between here and here is essentially right here. So if we were to look at this, everything up here is the matrix of the mitochondrion. Everything down here is the intermembrane space. And everything that is right here is the inner membrane. Okay? So what that is the matrix, the inner membrane, and the intermembrane space. Now these all have to be in the right sequential order to work. But these can be anywhere on here. So the beauty of the mitochondria, why does it work so well? Think of taking like a 55-gallon bag and sticking it in a small plastic grocery bag. You'd have to fold it up to do so. So this inner membrane is basically like a bigger folded bag than this outer membrane. And what that allows is a lot of surface area. So there would be tens of thousands or more electron transport chains on this inner membrane, all pumping H plus into this very small volume. The matrix staying full of fluid would keep this blown out to the side so that this volume stayed small, allowing your hydrogen gradient to build up more quickly so that you can make ATP more quickly. Having more surface area allows you to make more hydrogen or push more hydrogen into that inner membrane space more quickly, so that in letting that hydrogen get back into the matrix, you make more ATP. Now, the next question becomes, where does all of this NADH and FADH2 come from? Well, I'm going to try to draw that up in the corner. Hopefully I can get it all to fit, and hopefully you can see everything that I'm trying to do with it. And what I'm going to do is I'm going to start off just by drawing this six carbon molecule. Let's say that each ball represents a carbon. That breaks down through a process called glycolysis into two 3-carbon chains. Those 3-carbon chains are known as, this would be up here, this one is glucose. The 6-carbon is glucose. These are pyruvate. And what ends up happening to these is these will get fed into Krebs cycle if you're using Krebs. So glucose is what we start with, and we break it into two parts. That process is called glycolysis. Glyco means sugar, lysis means to cut. Now, in this process, two things happen. Two things are made essentially. Overall, what's released, I'm not worried about what's put in. What's released is right here. You take in 2 ADP, turn into 2 ATP. At the same time that you make those, you also have 2 NAD plus turn into 2 NADH. So basically what you're doing when you break this carbon-carbon bond, you're stealing those electrons and you're sticking them on these NAD pluses to make NADH, along with a few other electrons that are in high energy positions. Now once you get through this, all of this process between here and having glucose is glycolysis. Then this breaks down a little further, and we'll come back to that in a minute. And then it goes through what's called the Krebs cycle. And in the Krebs cycle, you end up with 2 carbons being fed in to a 4-carbon chain. Now that 4-carbon chain and these 2 carbons, as they combine, they're going to become a 6-carbon chain. That 6-carbon chain will start to break down into first a 5-carbon chain, and you'll see some familiar things here. The first thing to come out of this process, when you go from 6 carbons to 5, it has to come out as something. Well, it comes out as CO2. And anytime a CO2 comes off, you're breaking a carbon bond, the carbon-carbon bond. And the electrons in that bond are fed off as an NAD plus becomes NADH. Now, the next thing that happens is this 5-carbon turns into a 4-carbon molecule. That 4-carbon molecule means that another CO2 had to come off. Now that CO2, when it came off, it broke another carbon-carbon bond. So just like this one, what ends up happening to make that, what ends up happening with those electrons is an NAD plus was turned into an NADH. Now, if you've already noticed it, up here we said this was 3 carbons and this was 2. Well, there's another step called the intermediate step up here. I'll let you guys include it in Krebs cycle. This means that another carbon up here had to come off as CO2. Well, if a carbon came off up here, and we look down here, every time a CO2 came off, it made an ADH. So, as you probably guessed, to make that happen, you had an NAD plus break off as an NADH. So this NADH pulled the electrons out from between these carbons again. So down through here, this essentially accounts for that pyruvate being totally broken down. But that's not all the energy that comes out of it. The reality of it is, we keep converting this 4-carbon into a different 4-carbon, then a different 4-carbon, a different 4-carbon, a different 4-carbon, until we get back to this thing up here. This oxaloacetate that we've got up here, it's got to be in that form to combine with these 2 carbons and make this molecule called citrate. Citrate, or citric acid, is the reason for the other name of the Krebs cycle, which is the citric acid cycle. Now, as we rearrange these 4, what we keep doing is we keep giving off other molecules that are of value. So down here at the bottom, we actually have a substrate level phosphorylation. We have an ADP turn into an ATP. Then, right after that, we have 2 hydrogens that get broken off of the 4-carbon molecule, and those electrons are stolen. And at that point, let me actually use green for this. At that point, because we use green for it down here, an FAD and FAD turns into an FADH2. Now, finally, this goes through like 2 more conversions, and another hydrogen gets broken off up here, and we have another NAD create an NADH. Now, what that means is, in the Krebs cycle, we make 1, 2, 3 NADH. We make 1 ATP and 1 FADH2 per pyruvate. But if you want to simplify it, you can include the intermediate step instead as well, and you can say we make 1, 2, 3, 4 NADH, 1 ATP, and 1 FADH2 per pyruvate. but that's only one of the pyruvate per glucose. So if you wanted to look at this per glucose, you'd have to say we make 1, 2, 3, 4 times 2, because this pyruvate is going to go through the same cycle, so you make 8 NADH. You make 8 NADH. You make 1 times 2, because that's going to make 1, 2. You make 2 ATP directly, and then you make 1 in from this pyruvate. That pyruvate's going to make another. You make 2 more in FADH2. And up here you got glycolysis. The glycolysis part of this makes 2 NADH and 2 ATP directly. Now what are these worth if we instead convert them into all ATP? Well, when we convert NADH, that NADH, the way it works out is you have to take this to find out how many this makes. So this NADH, you have to multiply it times 3 ATP per NADH to get 6 ATP. You get these 2 ATP, so what happens is they stay 2 ATP. Now, the next thing we've got is these 8 NADH times 3 ATP per NADH equal 3 times 8 gives us 24 ATP. So that's a lot. This FADH2 equals 2 ATP per NADH. So this equals 4 ATP. And then we get this 2 ATP that stays 2 ATP. Now, the reason why NADH and FADH2 make different amounts of ATP is, if you'll look right here, NADH goes through, and I drew this as 6. I don't care how many you draw it as on your test, but I drew it as 6 so I could show you generally correctly what happens. If this pumped 1, 2, 3, 4, 5, 6, I made them red hydrogen to match where they came from. FADH only went down 2/3 of this, so they made 1, 2, 3, 4 hydrogen. Well, NADH makes 50% more hydrogen. It travels down the full electron transport chain. This one only goes down 2/3. So if you'll imagine this coming over here and it taking 2 hydrogen to get out to make 1 ATP, and that's not what really goes on in this system in real life. It's not a nice clean 1-to-1 ratio, but in general, NADH makes about 3 ATP per NADH. FADH2 makes about 2 ATP per NADH. So if we go back up here and we look and we say then glycolysis makes a total of 6 ATP from NADH plus the 2 ATP from straight ATP, we get 8 ATP in glycolysis. The NADH in the Krebs cycle or citric acid cycle, that's 8 NADH times 3 gives us 24. So we do 24 ATP from the NADH. 2 that are made directly, and 4 more that are made by 2 FADH2 times 2 ATP per, this should have been FADH2. That gives us 4, so we do 4 ATP. So what we have here is 24 plus 2 plus 4, that's all together, that's going to give us 30. 6 plus 2 gives us 8. That is our total of 38 ATP. Now, if you've been in class, you got a better graphic on this. I was trying to fit it all on one board, make sure that I had it done tonight so that you guys would have a shot at it. Anyway, when you're in tomorrow, I don't want you trying to memorize every single little thing. I want you to be able to draw this process and understand what's going on. I want you to be able to draw the mitochondria and understand what's going on. And I want you to be able to draw this cycle just with the outcoming stuff. You don't need any of these blue things in there. I don't care about the drawings or how many carbons at each spot. What I care is, you know, as CO2 comes out here, it makes an ADH. CO2 comes out, makes an ADH. CO2 comes out, makes an ADNH. You do that 3 times in a row. Then you go AFN, ATP is made, FADH2 is made, and ADH is made. That will allow you to count these up, know what glucose does. The only things I want you to remember is 2 ATP per FADH2, 3 ATP per NADH. That should allow you to use this process to help understand this and get this drawing right. This process will reinforce this one. This process will reinforce what goes on and where on the mitochondria. So this is what I'm looking for kind of overall. That is more or less the entire unit. The only thing that's not in here is fermentation. There's no space to include it, and I want to go ahead and get this up in the form that it is. So hopefully this helps you out some. I'll replace this with a better video later, but I want to get it up so you guys can see it.